The last thing that I want to talk about is regulation of the cell cycle. So everything that we have been talking about up to this point is really under cellular control. And it's not a one size fits all. The frequency of how often cell division, how often the cell cycle occurs, as well as the pace at which it occurs, differs depending upon what kind of cell we're talking about and depending upon how quickly those cells need to be replaced. So for example, if we look at this table, you can see something like a red blood cell that we use a lot of frequently is we replicate about 2.5 million per second. So it's happening very quickly and all the time. As things like skin cells as well, that you are constantly sloughing off, are always dividing and rapidly being replaced. But things like nerve cells, once they are born, never divide. Right? We can produce new nerve cells, but we do that through stem cells, not through division of existing nerve cells. Right? And things like our liver cells, sometimes when we have damage, will go back into that cell cycle and replace themselves. They will do that for repair. Okay, so if we go back and we look at this cell cycle, you can see that every point along the way, there are checks and balances to make sure that the process should still occur. So after mitosis, once a cell is born, it grows, right? G1 is its first growth phase, and this is where it's getting to its proper functioning size. When it thinks about getting ready to divide, it has to do a couple of checks and balances. And one of the first things that it does is look at its DNA. Is my DNA okay? Should I divide? Should I replicate it, right? Because you don't want to be perpetuating the problem. Once it goes through that S phase, then we're looking at, am I replicating my DNA correctly? Is everything lining up like it's supposed to? I get into my G2 phase, where I'm starting to do additional growth, preparing for cell division, making those organelles. I'm looking at things like, did all of my DNA get copied like it was supposed to? Is there any damages that I need to fix before I move on? Is my mitotic spindle functioning like it should? And if all of that passes muster, then we can go into the division process. And it's not a all or nothing there either. When we get about halfway through, we stop at that metaphase. And before we divide up our chromosomes, we want to make sure that everything is like it should be. Are all of my chromosomes attached to a mitotic spindle? Do they align down the center like they're supposed to? Because this is a really crucial point where I'm separating out my chromosomes, and I want to make sure that everything separates and I get equal division to the two different sides to my new daughter cells. Okay, so there's all these internal checkpoints that the cell does. The other thing I want to point out in this cycle is one last thing right here. This is the G0 phase, and we refer to the G0 phase as the resting phase. So when a cell has gotten to its proper functioning size, especially in an adult individual, it's going to go into the G0 phase, the majority of our cells, because we're not continuing to grow, we're not doing any repair, we're just doing our job. Okay? And so some cells come into here for a period of time and then go back into the cell cycle. Some, like those neurons that I was mentioning, come out into that G0 phase and they never go back, right? So you can go back and forth between these, but as an adult, the majority of your cells are in this phase, in that resting phase. Okay, so if we think about these kind of checks and balances and whether or not things are going to move forward or stop, a lot of it is controlled by internal signals. And an example of this was that one I was talking about in metaphase. So let's say we get our chromosomes and they're lined up like they're supposed to, but the kinetochores are not attached to the spindle microtubules. And so that sends out a signal that says, whoa, don't release that separase enzyme to break those cohesins because we're not ready yet. And so it will delay that process moving forward because we're not set to where we should be. That machinery isn't functioning like it's supposed to. And there are numerous different chemical signals that happen internally within the cell, and they fluctuate. We refer to them as cyclins because they're cycling their abundance during different phases when they are needed and when they are not. Okay. But it's not just internal signals that influence the cell cycle. There are other things that go on that are external to the cell that are really important to influence whether or not a cell is even going to divide. And one of those are growth factors. Growth factors are released by certain cells that promote other cells to divide. And one really important one is this PDGF, the platelet-derived growth factor. And so one of the things that we found to be very difficult when we started looking at cells in vitro, so in a dish as opposed to in real organisms, is that it was really hard to get them to divide if they didn't have the proper external signals, right? So remember, all of your cells have these receptors, these protein receptors that are embedded in the plasma membrane. And some of them act as signal transducers, right? So when that growth factor is present and it binds to that receptor for cell division, it starts a cascade of events that basically causes that cell to divide. If it's not there, then the cells basically are in stasis, they're waiting. And when we think about normal cell division, they have a couple of other factors that influence whether or not they are going to divide. One of them is anchorage dependence, right? Especially in our multicellular organisms, right? You have to have that extracellular matrix because you are multicellular. You need to have communication between cells. You need to have connection between cells. And if you don't have that, then the cells don't know what's going on, and so they don't divide. Right? The other thing that normal cells have, is density dependence. And the reason they have this is because if you have too many cells, then there's not enough resources for all of those cells, and you start to have cell death. And so when those cells divide, they basically push adjacent cells out. So you get that single layer of cells. If you remove some cells, then division will continue, right? So you can see here, at this point, we have anchorage dependence. Those cells start to divide. And then once you form that single layer, it stops. But if you remove some cells and you have more space, then the cells will divide again. Okay. Now, the reason for this, again, is because in a single layer, you have a huge amount of surface area for resources to come into those cells. So things like oxygen and glucose. This doesn't always happen, though. Sometimes we get cells that they basically make these masses where they overlay on top of one another. They ignore those regulations, those external regulations. And that's when cancer happens. That's when you get those ignoring of the signals and allowing errors to perpetuate. Okay, so you can see there are many checkpoints, signals, internal and external, that control whether or not we will proceed, right? And this is for production of new cells. But it's not just production of new cells that's under cell control. It is also cell death. And cell death is referred to as apoptosis. Apoptosis is basically when the cell says, this cell, this cell, this cell, they should be eliminated. Okay, it's a natural process. And this is something that is really important, right? So think about during development. In utero, you all have had webbed feet and webbed toes. But at some point during development, you can see here these yellow dots. Notice where those yellow dots are migrating into the area between the digits. Those are the cells that are basically marked for death. And so over time, those cells get eliminated. So then you end up with those individual fingers and toes. Okay. So when we think about how apoptosis is occurring, apoptosis basically results when there are proteins within your cell that basically say, don't self-destruct. But if you get a marker that says, whoa, go ahead, then the cell will basically go through and destroy itself. Okay, so let me show you what I mean. So here we have a normal cell, and out here we have a death receptor. Okay. If in the normal situation, we have this protein present right here that inactivates or that inhibits activity of these CED proteins. And those CED proteins are basically the ones that start this cascade of destruction. And so right now in a normal cell, they're basically inactivated. Okay. But when I get that death signaling molecule attaching to the death receptor and these executioner proteins, basically what happens over here is that the activation, the cascade of events, keeps this molecule, this ED9, it inactivates it, which means that these executioner proteins basically are given free rein to go ahead and start destruction. But it's not like a complete disorganized process. What happens is production of these structures that are called blebs. And blebs basically you're taking that cell and you're partitioning off into little vesicles, which basically are called apoptotic bodies. And then you have the lysosomes come and fuse with these to break the stuff down. So it's a very orderly process. And it's a very important process, right? This is how your body controls errors that are produced. And also, you know, the whole developmental component to it. But this is how you prevent errors from occurring. And really, when I say errors, I mean cancer. Right? Cancer is when your cells are not listening to those signals, those normal signals in the cell cycle, and they go out of control. So when we think about cancer cells no longer have that density dependence. That's how they make the tumors, right? They start to build on top of each other. They no longer have anchorage dependence. They don't respond to those normal signals, right? They say, oh, my DNA didn't get replicated. Oh, well, I'm going to keep going. Or, oh, my DNA didn't separate like it was supposed to. That's okay. Let's keep going, right? In addition to that, a lot of our cancer cells that we have found suggest that they don't need external growth factors. So there's very little regulation in terms of either external or internal that are saying, don't do this, you're making an error. Okay? There's a link here to a video that kind of compares the speed at which cell division occurs between normal cells and cancer cells, and I would highly recommend that you watch it. The other thing that I want to mention here is something that I think is important to think about when we talk about cancer cells. One of our very first ability to look at tissue culture, right? One of the things that I said, tissue culture is kind of hard because you need to have those external signals. You have to have density dependence, you have anchorage dependence, all of that stuff. So it's really hard to get cells to grow in tissue culture, right? They're very finicky. And not only that, they don't live for a very long period of time. So it was a while before we got our very first immortal cell line. And those cells came from a woman named Henrietta Lacks. So Henrietta Lacks was an African-American woman in the 1950s. She had a very aggressive form of cervical cancer. So she went into the hospital and was evaluated. And some of her cells, both the cancerous cells and her normal cells, were cultured. And the cancerous cells were ones that were very intriguing. So this physician, Otto Gay, what he realized is that over time, these cells just kept dividing and dividing and dividing and dividing. And he said, this is great, right? Because now I have a line of human cells that I can use for research, and they're not as finicky as those normal cells that we were using, so we can continue to research them. And so those cells were sent worldwide for biomedical research. They were used for the polio vaccine. They were used for HIV treatments, numerous pharmaceutical drugs, makeup, various things, right? Over 15,000 patents were made off of tests done with those cells. Now, the interesting thing about this is that at the time, there weren't any HIPAA regulations. So she wasn't informed, nor was her family, that her cells had been taken and that they had been sold all over the world. And so in the 1970s, the scientists were starting to, put things together more, and they were like, I wonder why these cells are still dividing. If only we could get some more DNA to look at, normal DNA, because obviously something is wrong with her DNA because the cells are not listening to the cycle. So if we could only get some real DNA, but she died. She died shortly thereafter in the 1950s due to complications associated with the cancer. I think it was blood poisoning. And so, well, she was dead, but you know what? She had kids. And so in the 1970s, her children and her family started getting calls saying, can we get a sample of your DNA? Because we want to know why your mom's cells are still dividing. And they're like, what? Remember, I said 15,000 patents were made off of drugs based off of research done on her cells. That's a lot of money. And none of it went to the family at all. And so when we think about this process here, they didn't have to inform the family. This is part of the reason why those HIPAA regulations are so important and why you should read the fine print when you go to the doctor's office. Right? Her family was extremely poor, so poor that she never even had a headstone. but all of the proceeds from the drugs and the treatments and all of that kind of stuff that was made based off of research on her cells, the family saw none of it. Okay, so it's an interesting story. Over time, her family became, they got okay with the situation, even though they saw no proceeds from it, because they realized the fact that even though in death, their mother was making a huge contribution. Okay? So if we think about what we learned from her cancer cells, we realize that cancer cells may not need growth factors. And in fact, some studies suggest that some cancer cells are able to make their own or act as if it's present, even though it's not. In addition to that, you have these normal cells, right? These normal cells that ultimately are transforming into cancer cells because of errors in their DNA. And because of the fact that they no longer listen to that whole density dependence, they make these tumors. That's where your cells are basically continuing to divide, and instead of spreading out into an even layer, are making these bumps, these masses, right? And when we think about cancer, there's two different types of cancer. There's benign and malignant. And the main difference between them is benign remains at its site, and it remains at its site because it has a sheath around it that kind of contains it. Right, so these would be your lumps, right? And even though benign are not spreading, they can be very detrimental. Think about people that have those huge benign tumors that interfere with their ability to breathe because it's on their face, or to swallow because it's in their throat, right? So just because it's benign doesn't mean it's not bad. Right? The other type of cancer is malignant, and malignant is a problem because it travels and it infects adjacent tissue. Right, so if we think about how it does this, if we have a cancer that is malignant, the main difference is that it doesn't have that sheath around it. It doesn't stay isolated or located in one spot. And so individual cells can break off, and they can break off and get into the blood vessel or into the lymphatic system, where they can be transported throughout the body. And then they basically invade adjacent tissue in a new location and make secondary tumors. Right? And so when we think about cancer, we talk about, let's say we say you have breast cancer. It's called breast cancer because it started in the breast, but that doesn't mean it stayed there. It may have metastasized and spread to other locations, resulting in additional problems. So ultimately, when we think about cancer, cancer is an error in the mitotic process. Cells are not listening to the signals, and so they're going through and perpetuating the errors, and which ultimately results in disease.